Well, thank you everybody for joining us on this webinar. We plan on putting more together of this type of information presentation as we go forward. So thank you for joining us today. I'd like to introduce myself and Narek, my coworker here. I'm David Cook. I am a business development manager for the West, one of the sales reps here. Narek is our chemistry and operations supervisor. He'll be going over some of the technical aspects of the test here shortly. For certified laboratories, which we actually specialize in cosmetic and OTC testing. So we appreciate your time. Again, Narek will go through some of the chromatographs, the matrix, and data analysis review, which I think a lot of you are looking forward to. So today I'll highlight why 1,4-dioxane testing is becoming essential for compliance and brand protection. And at the end, we'll answer some of your questions during a Q&A session. So hold on to the end. We'll go ahead and be addressing some of your questions at the end of the presentation. Next slide, please. So what is 1,4-dioxane? 1,4-dioxane isn't added intentionally. It's actually a byproduct of surfactant manufacturing. I believe it's called ethoxylate or ethoxylation. It became a hot topic when it was found in New York, Long Island groundwater, triggering regulatory scrutiny. New York was the first to set strict limits in the US. Right now, I believe that's, as I have it, 10 parts per million for cosmetics and 1 part per million for personal care products. And that was established in 2023. We do expect other states to follow as testing sensitivity and awareness grow. California has already joined that. And again, we think more will be following suit. Next slide, please. Why 1,4-dioxane matters? It really comes down to we believe it's a carcinogen. It's a human carcinogen. So that's the real root of the issue here. So it's not really all just about compliance and meeting state needs. It's really about protecting consumers' trust and really your brand reputation by proving that you don't have this solvent or concern in your product. Next slide, please. So why are we highlighting this? There are some challenges in detecting 1,4-dioxane. There is a traditional USP method that's been around for a little bit, 467, or even the EPA 8270 method. These methods don't have a low enough detection limit based on the state's strict limits they've set, and these methods struggle with cosmetic matrices. So that's really the issue and where we come in. We find that products with oils, surfactants, or even emulsions, they create interference and it can hide that trace contaminant. So let's get into a little bit about what we've done on our side. Next slide, please. So Certified Labs has a GC mass spec solution. We've actually have a validated GCMS method that detects down to 0.5 parts per million. That's 0.5 parts per million, 20 times more sensitive than the current regulatory limits. To put that in perspective, the USP method I mentioned a little earlier, that LOD or limit of detection is at 380 parts per million. So a big difference, right? So our validated method was designed for liquids, creams, and gels. So really the products that typically would have this type of concern and a lot of the products that we deal with at our laboratories. So your results are accurate and defensible, and we'll get into some of those details with Narek here in a second. Our turnaround times are fast and consistent at the lab, really ideal for batch release or reformulation monitoring. So let's go to the next slide. Why independent testing matters. Suppliers' COAs rarely even test for 1,4-dioxane, or if they do, they might be using that USP method, and the testing limits are just too high to be usable. So independent verification protects against recalls, penalties, and really ultimately loss of consumer confidence. So with that, let's get into some of the real details here of our method and 1,4-dioxane testing. So I'm going to turn that over to Narek, and he'll continue with this on.
All right, thank you, David. Yeah, jumping more into the instrumentation side and the technical side. So, 1,4-dioxane traditionally, with methods such as the USP method, has been run by GCFID. And the problem with that is that FID relies on retention time and column chemistry to sort out and detect dioxane. This can often cause false positives due to interfering peaks. And on the top right side, you can see the chromatogram of the USP residual solvents method for the standard containing dioxane. You'll see that the 1,4-dioxane peak is actually really small with a low area count and a low signal. So the advantages of using GCMS, such as with our method, are that we have a much lower detection limit and we have much higher area counts versus the FID. We also use a headspace injection to isolate volatile solvents from other components in our sample formulation, giving us a cleaner analysis and preventing contamination and interference. And our MS detector lets us scan for target ions and really focus in on detecting dioxane. The way this works is each compound we detect produces a unique ion signature, and we can compare these signatures to the spectrum of a dioxane standard. This helps us confirm if a sample really contains dioxane or just has interfering compounds. So on the bottom right, you'll see the typical mass spectrum for dioxane. Next slide, please. So our method works with a wide range of finished goods and product types, including but not limited to shampoos, sunscreens, lotions, and we achieve this by using a targeted extraction style for water-soluble products and water-insoluble products to really help us pull any residual dioxane out of complex matrices. And in the video, you can see our Headspace autosampler preparing a sample for injection. Next slide, please. All right, now I'd like to get into some of the chromatograms and data. So on the top left in the blue, you can see a chromatogram of our 1,4-dioxane standard. It's got a clean peak with no noise in the region. And to the right of that, we have two other chromatograms scaled to the same level as each other. The middle chromatogram, once again, shows our dioxane standard peak with the retention time around 3.5 minutes. And to the right of that, we have a sample chromatogram. So you can see in the sample chromatogram, even though at the later retention times, there's other compounds being detected, what matters is that at the retention time of 1,4-dioxane, which is around 3.5 minutes, there's no peak being picked up. So this sample is dioxane free. And on the bottom left, you'll see our linearity curve. This is a plot of our dioxane standard at different concentrations versus their area counts. So when we test a sample and we actually detect the dioxane peak, we can plot its area along this curve and quantify the concentration of dioxane that is present. And to the bottom right, you'll see some data from our spiking studies, where we had two operators prepare replicates of a spiked sample, and their recoveries have low IRSDs, so that confirms the precision of our method. Next slide, please. So after testing is completed, we'll issue a C of A, and we have two types of reports. Either you'll get not detected less than LOQ, which in this case, the LOQ is 0.5 ppm, or if dioxane is detected in the sample, we'll report the quantity of dioxane either in ppm or percentage based on the tester cost. And to the right side, you can see a sample C of A with the not detected result. Next slide, please. So the methods we perform at certified labs are scientifically justified. We go through validation where we follow FDA and ICH guidelines. A lot of our methods are also ISO accredited, meeting their standards. And when we validate a method, we run a series of tests to prove the reliability and robustness of our method. We ensure the method is specific to the analyte we're looking for, in this case, 1,4-dioxane. We ensure the response of our detector is linear, So as the concentration increases, the signal increases as well. And we ensure that the methods are accurate and precise and robust, meaning we can have multiple different analysts perform the method, multiple pieces of equipment, or even between different lab locations. In addition, this ensures continuity of the business if we encounter problems with a piece of equipment, for example. So in summary, we've developed a new method for 1,4-dioxane with lower detection limits than more traditional methods. And our new method meets the modern requirements that are set at a state level. In addition to New York and California, more states are now starting to set restrictions on these impurities and residual solvents. So not just 1,4-dioxane, but also including benzene, methanol, diethylene glycol, ethylene glycol, all of which we provide testing for at Certified Group. And I'll hand it off to David for the rest of the slides. Thank you.
Thank you, Narek. That was insightful. We appreciate you getting into some of that detail. There may be questions that come up again at the end. We'll be covering some of those questions. So kind of closing this out here from us. Certified Laboratories, again, we have this GCMS validated method down to 0.5 parts per million, really low for the cosmetic and personal care. We have 4 labs in North America to test. We have PhD level chemists with a deep experience in cosmetics and OTC testing. We do offer other contaminant testing too, obviously, things like residual solvents, heavy metals, and pesticides we can do. And there's even other contaminants that are coming down the pipeline that we have methods for, or always watching what's changing so we can look at new or developed methods ourselves to help satisfy these testing requirements. So our goal here is to really help your compliance be simple and proactive through fast and efficient validated methods. Next slide, please. This slide just shows a little bit of where those labs are located. We have Burbank, California. We have Carrollton, Texas. We have Melville, New York, and we actually have Edmonton, Canada as well. Those are our North American labs for our OTC and cosmetic testing. Next slide, please. So feel free to reach out to us to discuss any testing needs, onboarding process, pricing, things like that. We can align our testing with your quality systems to really help design an efficient compliance program to help you get through some of these requirements. Next slide, please. Go ahead and go to the next slide, please. That might be the, it just shows a little bit of our capabilities. We have over 5,000 tests offered at our labs, so pretty robust and extensive. This just shows a little bit of them. Some of the new ones like colloidal oatmeal, a lot of labs can't test for, we do. Benzene, there's a host of them here you can see, formaldehyde that we offer here at our labs. We do a lot of SPF testing as well for the OTC side of it. Next slide, please. This just highlights some of our team on the sales side. Well, the president, he oversees everything, Todd Shea. The VP of our sales and marketing, Stephen Tarmino. Our account reps, some of you may be working with. We have Maritza, Michael, myself, Carla, Alan, and Ashley. So just like to highlight my team. They're very helpful. So if you're working with any of them, you can reach out to them directly with any questions about some of this testing or services. Next slide, please. So Q&A. Before we get to the Q&A, at the end of the seminar, we'll be sending you a post-webinar survey. When you actually close out the webinar, there'll be a quick, brief 6-question survey. That's going to help us, really, if you could take the time to fill that out. It will help give us feedback to better understand what you're needing 1,4-dioxane testing for, possibly address any remaining questions that you might have from the webinar as well. So we appreciate your time. If you could just fill out, again, it's a 6 brief question survey at the end. So let's get to the Q&A. We did get some submitted, and we'll look at some of the ones you're typing in as well. I know one big question everybody asks is, what do you charge for your 1,4-dioxane testing? I mean, obviously that's important. Our current pricing is 175, so it's $175 per test. We typically ask for a 20-gram sample, and our standard turnaround times are 7 to 10 days at the moment. If you had a need for rush testing, just reach out to us. We can see what we have available and quote that out for you if need be. I know, Narek, I think you had a couple of technical questions as well that were kind of pre-submitted that we could cover real quick.
Yeah, I think there was one question about which class, which categorization dioxane falls into. Um, so I believe dioxane is part of class two residual solvents, and it has a per USP, it's a 380 ppm limit, which is what you explained, David. But again, the main issue is that most states are starting to update it to much lower limits. Like you said, it's either 1 ppm or 10 ppm, depending on the type of product.
I see some good questions submitted here. I'm not a quality person, so let's see. Well, I know this is recorded. I believe this presentation would be available for any of you joined late that you'd be able to see. So yes, I think that is a yes. So that's the good news there. The question on 1,4-dioxane in New York per batch, or is it per formulation? Do we, I don't know, have you seen that, Narc? I don't know if we know that from their standpoint, that they might be outside the scope of what we typically do on our side. Um,
So if
I'm
the question is, are the limits are per formulation, but how frequently the testing needs to be conducted, I would defer to quality for that. I think we can get an answer on that from quality and send a follow-up.
Yeah, it's a good question. It's really kind of outside our scope. I would believe that it would be somewhat related to batch if I was taking at it. I don't, you obviously want to test your formulation. That would give you the key, but there may be some possible back end batch testing they would want as well to confirm it's not popping up anywhere. But yeah, it's a great question. Um, so there's a question, I think, do we plan to update the existing residual solvent panel? As we see those changes come through, we would potentially be adding these to some of the panels. I think these are currently separate tests, but as we start to see class one, two, and three possibly being updated, that's something we would definitely be looking at considering as part of a full panel test for the residual solvents. But at this point, if you need this test, it is a separate request.
There's a question about which class it falls into again. Yeah. Class one, I think, is only 5 solvents right now, one of them being benzene. So dioxane is class two. And I think like the classification also determines the danger level. So class two being intermediate to going more into dangerous. So
Johanna had a question here. It's a good one, really. It's the difference between the EPA or even the USP method versus our validated method. It's really the LOD or limit of detection. That's the biggest difference. Um, like, I'm not sure if EPA's LOD, I don't know if it's the same as the USP, but USP is 380 parts per million. Our validated method goes down to zero point 0.5 parts per million. So below the state's requirements for testing, where the USP method and I believe the EPA method are just too high. You're not going to get a read on it based on what the states are asking for, at least for New York and California at this point. Lisa, you had a good same similar question on frequency of doing the testing. And that's a QA question. We can see if we can get some back-end information to support. Not 100% sure on the QA requirement for is it batch production yearly, every 3 batches. I don't, I, yeah, sorry, don't have that specific question, we'll try to follow up with an answer on that.
There's a question here about the method is validated for cosmetics, and do OTCs fall into that category? Yes, yes. So they do fall into the category, and we do regularly test sunscreens, acne products for dioxin.
Yeah, it's a good question. If something odd came through that we might question outside of that scope, you can reach out to us. We can have the technical people like NARC look at it and see if they think there's going to be any issues with it. Um, maybe is it an acne patch? That would be one I would think of that I would want the team to look at. They may have already seen it come through, but those are the kinds of outliers, typically outside of the oils, the creams, the lotions that we typically see this issue potentially pop up on.
And we do have a feasibility process where if it's some sort of matrix that's just uncommon, we can attempt a feasibility to see if it does work with the method, if it's being properly extracted, and give feedback on that to see if it's eligible for testing or not.
Yeah, there's a question on what kind of products we typically see this in. It was covered a little bit earlier, but it's products that have usually surfactant manufacturing, um, foaming, those types of things like some soaps, shampoos. I- It's a process called ethoxylate or ethoxylation.
Yeah,
You get
most products
surfactant.
that have like polyethylene glycols or polysorbates in them, they add ethylene oxide to exactly try to make it more foamy and more help with the feel of the formulation. And then as a byproduct, dioxin is performed. So it's unintended, but that's where it comes from.
Jitendra, nice to see you on here. I see a question. It's not more than 10 parts per million. Yeah, that's why we developed this method really is we want to be able to test below that limit, or if that ever changes. So yes, that's exactly why we're letting you know that this method exists, because USP methods and EPA just aren't going to help you. You can get a report, but it's not going to tell you if you're meeting these state requirements, and we can. So that's the good news, and it's a fully validated method that we've been testing for a while now. This is not brand new. We've been testing it for several months now.
David, click just one more time to clear it.
Thank you.
Yeah, there you go.
Yeah, Victoria, we can look at that. We can always talk about that online. Your question is basically, in the past, class two solvents have been tested. Prior to us developing this method, we only really had the USP method, the EPA method. So we were doing USP testing in the past for 1,4-dioxane. But again, with some of this new scrutiny that's come about, we needed to develop a method that has the lower LOD, and again, that's what we have here. Similar to some issues you see with Prop 65 for California, some of these requirements they're putting on brands and manufacturers are below what labs can test for. So labs are playing catch up to try to come up with methods, develop methods to test down low enough to give you some accurate, defensible result. So, sometimes states implement these rules and guidance that's, ahead of the labs. So it's a challenge, and we're trying to keep up with it. But it's a good question. If you need more on that, Victoria, just reach out to us. Let's see. I think we did that one. I think we've covered a lot of these. Yeah. If anybody has any last minute questions, feel free to type them in there. Again, this is being recorded, so it should be available to you to review if you'd like. Feel free to reach out to us directly if you have any questions. add our general number, and they'll put you in contact with one of us that can answer your questions. So again, at the end, when you close out, there'll be a brief 6 questions survey. If you'd be kind enough to fill that out, it is very helpful for us. We do plan on putting more educational, these free type of webinars available to the public. So watch for communication on those. We hope you found this to be valuable. Narc, thank you for your time. You're an expert in this area, so we really appreciate your time. Other than that, thank you for joining us today.